Well, good evening, everyone. I'm Dr. Britt Nicholson, Senior Vice President for Development at Massachusetts General Hospital. And I'm delighted to welcome you to our virtual symposium tonight. Well, first of all, thank you for joining us. Normally, you'd be greeted by our president, Dr. David Brown, but unfortunately, David was unable to attend this evening due to an unexpected death in his family, which occurred yesterday. Now, I look forward to this event every year because in so many ways it is a celebration. It's an opportunity for us to laud you, the donors who keep us going and who sustain us, to give you a glimpse of the good that you make possible through your contributions, and to say thank you sincerely on behalf of our patients and staff for your continued support of the MGH Fund. In addition to the MGH Fund supporters who are joining us tonight, I'd also like to thank the members of our Phillips Society for tuning in. You've chosen to include Mass General in your estate plans, and in turn, allowing us to continue to plan for the future. The future and our ability to respond is something that's always on the minds of two of our featured speakers this evening. And tonight, you're going to have the chance to hear from Dr. Ruan Barnavus, our new Chief of Division of Infectious Diseases, as well as Dr. Eric Rosenberg, Director of the MGH Clinical Microbiology Laboratory. And they'll be discussing the power of partnerships across the hospital and how these ongoing collaborations help us achieve our goal of providing the best patient care possible. Now, the MGH Fund represents our philanthropic bedrock. And what do I mean by that? Well, from patient care and research to education and global and community health, the unrestricted dollars given by donors like you provide the stable support that we need to maintain and drive nearly every element of Mass General's mission and operation. You know, the flexible funding made possible through the MGH Fund enables us to respond to unexpected challenges as well as meet unexpected opportunities. It allows us to seed innovative programs and invest in talented clinicians and scientists. In fact, your contributions ensured that we could react and respond quickly to the emergence of COVID-19. Your contributions kept us going through the ups and the downs in the months that followed. And now they're helping us ensure that we are prepared to lead the response, not only to future unexpected health crises, but also to exploring new ideas and opportunities, which allow us to continue to stay at the forefront of medical innovation. When I look out at the faces on my screen, I'm not just looking at the faces of grateful patients, of family members, determined friends and coworkers. You are essential allies in our work, our partners in progress. And the partnership we have is powerful. It can overcome all kinds of adversity. It can drive the discoveries that will change medicine. It can raise the bar in patient care. And it can change the face of healthcare education and training. It can build healthier communities. And as you are about to hear from our special guest presenters, ongoing collaborations across the hospital enable us to remain nimble and keep ahead of future health pandemics and emergencies. So again, thank you. Thank you for your gracious support. Thank you for your generosity. And thank you for your partnership. Together, we'll shape the future of healthcare for our friends and neighbors, for our communities, and actually for the world. Now, you'll have the chance to ask Dr. Barnabas and Dr. Rosenberg questions later this evening during the question and answer portion of the event. And so a few housekeeping items with regard to the Q&A. I want to remind everyone in attendance that you do not need a microphone or video capabilities to participate. At any point during the webinar, you can ask questions, and you can do it anonymously if you prefer, through the Q&A feature. We're also recording tonight's presentation, and a shareable link will be provided after the event. And now it's my pleasure to introduce our speakers for this evening. Originally from South Africa, Dr. Ruan Barnavus is a distinguished physician scientist who has devoted her career to the treatment and prevention of HIV and other sexually transmitted diseases, and more recently, on COVID-19 prevention within households. The ultimate aim of her work is to identify effective and scalable infectious disease treatment and prevention strategies to increase access across diverse communities and promote equity in health. Before joining Mass General in January of this year, Dr. Barnabas was a professor of global health and medicine at the University of Washington. Now, in addition to Dr. Barnabas, you'll hear from infectious disease specialist and pathologist, Dr. Eric Rosenberg. Now, pathology is that branch of medicine that specializes in the diagnosis of human disease. It really is the invisible engine of every hospital, and that's never been more evident than during the COVID-19 pandemic. As director of Mass General's Clinical Microbiology Laboratory, Dr. Rosenberg oversees the scientific team responsible for diagnosing infectious diseases, everything from influenza to HIV. And at the height of the recent Omicron surge, Dr. Rosenberg's team performed between 1,000 and 1,500 COVID tests every single day. And now it's my pleasure to turn over to Dr. Barnabas to get us started. Ruanne.
Thank you so much, Dr. Nicholson, for that very kind introduction. I am thrilled to be here at MGH and honored to be talking to you this evening at the 1811 Society. Today, we're going to talk about the power of partnerships. And in my discussion with you, I will review how we are a global community connected. And that's certainly something that we have been aware of during the COVID pandemic quite acutely. So I'm going to talk about the power of partnerships and make 3 points. I'll share the pandemic journey with you domestically and globally, and how that helps us think about how we can be prepared for future pandemics. I'll talk about how we are connected globally and how we learn from each other. And then the ultimate aim of our work in infectious diseases is to do lab to patient science, to be able to translate very quickly what we do in the laboratory to having impact on patient care. And through this evidence-based framework, we hope to both strengthen science and decrease disparities in health outcomes. So let's start with looking back at our pandemic journey. And this slide in the background, you can see a picture which is actually currently the coverage of COVID-19 vaccines globally. And you can see there are certainly many countries in Africa and in Asia where the coverage of vaccines still falls behind. So let's go back thinking now this is globally the numbers of cases and deaths, more than 6 million deaths globally from COVID-19 from the start here in February. And I'm looking at the slide to make 2 key points. One is that we are in a very different place now, May 2022. We have highly effective vaccines. We have the ability of widespread testing. And actually you heard how many tests are done right here at Mass Gen in Dr. Rosenberg's lab. And we also have options for treatment for COVID-19. But let's look back to where this started in December 2019, March of 2020, when we had the first death here in the US and the first cases of community transmission. We knew we were dealing with a highly transmissible virus with increasing hospitalizations and deaths, but we didn't have the capacity to even know what our denominator was at that time because we didn't have the capacity for testing. We didn't have vaccines or treatment. And over this time frame, we have made great scientific advances. I think we want to be able to replicate this story in the future for future pandemics. And so I'll just draw attention here to December 2019 to March 2020. And just draw attention to this time. It's something we should reflect on. How could we compress this time frame, and if we were able to compress the time frame through which we could identify cases, would we be able to compress the overall time frame? And certainly, while our production of vaccines was groundbreaking in the speed in which we developed highly effective vaccines, as scientists and clinicians, we always want to think about what we can do to make that to have faster impact on health and prevent morbidity and mortality. Okay, so now I'm going to look globally. Here we are in the US, almost 1 million deaths from COVID. Here in Massachusetts, we have continuing surveillance and response. We are closing gaps in vaccination, testing, and access to treatment. But we're also here at this unique time when we should think about how we can prepare for emerging infections, and also, very importantly, how we build our workforce for infectious diseases. And this is in contrast to where I started my COVID-19 journey in Washington State in March of 2020, where we had the first cases of community transmission, the first death, and we were able to leverage our clinical trial infrastructure and work closely with labs. And when I reflect on what really drove our response to COVID, the fact that we had highly trained people who had experience and expertise working together, and that we were able to work very closely with our colleagues in the laboratory, meant that we were able to quite quickly assess the effectiveness of drugs to prevent the transmission of COVID. And so these relationships and this expertise is something that we want to continue to build moving forward. And the second point I want to talk to is about lab to person science. And there are really three aspects for this. One, clinical care, and then to leverage the clinical care we provide, that very special place we occupy right next to the patient. Think about how we can bring that quickly to the lab to identify novel organisms, and how we can build a workforce so we have people who can fill all these important roles. So the first, for clinical care, we think about how we characterize clinical cases, strategies to limit transmission within hospital and other clinical settings, share what is working, and we did a lot of this very well in COVID, and in the context of providing patient care, test new approaches, and we can do this through our clinical trials unit and other settings to do evidence-based research, empiric research. Working very closely with patients means we can also pivot and have a very close working relationship also with the laboratory to identify novel organisms, and have the capacity to rapidly identify and sequence organisms. We also have the option for testing treatment options in vitro in the laboratory, and this can drive clinical care and science towards interventions to prevent infection and treat infection. And then lastly, we always want to have an eye to the future, thinking about training and leadership. As we collate and synthesize data, we can impact patient care, we can lead clinical trials, and through both of these activities, doing science, leading clinical trials, we can train the next generation of physician scientists who will help lead the way in the future. We are so privileged to be at this academic hospital where we had early action for the pandemic. We were able to provide a rapid response, and for this it is essential to have people and facilities in place, and for people to have expertise and experience and a history of collaboration. And with these people and facilities in place, we have the capacity to expand our work. In addition, organizations need to be strong and flexible. And that is something that I have really loved about MGH since I have arrived, the innovation and speed at which problems are solved. And then thinking about our global partners and these collaborative partnerships to strengthen our pandemic response. And I'll just give an example of how we are a community connected globally. And I'll do that by talking about the Omicron variant, and that detection very early, first in South Africa and Nigeria, Botswana, and then later on in the United States and in Europe. But because we are a global community connected and we had strong collaborations with other scientists, over days and weeks, we understood that the Omicron variant was highly transmissible. It was likely to become dominant, that vaccination prevented severe disease and boosters increased immunity. And we were able to do that quite quickly, a more compressed timeframe than we had seen previously. And we were able to leverage clinical, laboratory, and epi data for this. And so this is also showing the numbers of confirmed cases and then much lower hospital admissions and patients in ICU and deaths with Omicron here, very different from the earlier waves, the Delta wave and previous waves that we had seen with COVID-19. And these data, understanding that they're different, helped us prepare for a different type of surge with remote care and telemedicine for oral antivirals. And we want to build resilient systems. And we know from HIV that we can build resilient systems that can be flexible according to the type of epidemic or the type of variant and its clinical course in a particular point in time. So as I wrap up, I'll leave you with these thoughts about the opportunities we have for preparation now. We can leverage our clinical environment and environmental data to serve patients as we are doing with, for example, the wastewater monitoring, but also our clinical monitoring as well. We can have systematic surveillance to fill laboratory gaps for emerging infections. And this is a priority for us. And we want to strengthen our global scientific collaborations and share technology. And importantly, we want to build a workforce for tomorrow through training and leadership today. Thank you so much for your attention. I'm very happy to take questions, and I think that will be after Dr. Rosenberg speaks, but I really appreciate the opportunity to share some of my reflections with you about where we have come from, what that teaches us about our relationships with each other, and what we need to do to prepare for the future.
So, Dr. Barnabas, I think you have to stop share. Okay, perfect. Okay, can you all see my slides?
Yes, we can.
Great, okay. So welcome everybody, and thank you so much for giving both of us a chance to tell you a little bit about our experience and collaboration during COVID-19. I knew Dr. Barnabas was going to take a global approach. And so what I really want to do is tell a little bit about what happened on the ground here at MGH and the lessons that I personally have learned along the way. So when I asked my wife in preparation for this talk, what lesson do you think is the most important? She said, never lose your sense of humor. And so throughout the entire pandemic, I always repeatedly said, never lose your sense of humor. But tonight I really want to share with you 5 specific lessons. But before I get into these lessons, I do want to send a personal thanks to all of you, because when it hit the fan here at MGH and my laboratory had to mobilize at a speed that we've never had to work before, I was able to go to the highest leadership at MGH and ask for things. And without any question, I got whatever I needed. And that was really because I think our leaders had the opportunity to rely on you as donors, and they had this pool of money that was able to be allocated for this kind of emergency. And so that, if there's anything I can say tonight, is really thank you, because you all really got us through the hard times. So the first lesson I want to share is from Oscar Wilde, where he said, expect the unexpected. And so in our business, pandemics and bio threats aren't new ideas. And in fact, in my over 20 years here, these are the things that we had to deal with on a regular basis. We had to be prepared for things like anthrax when white powder started showing up in envelopes. We had to be prepared for monkeypox when smallpox was a serious threat. And believe it or not, today we are currently ruling out a patient in our emergency room who is suspected of having monkeypox, which is a viral infection that looks for all the world like smallpox and is highly infectious. The big difference is we've always been prepared for these, but we really never expected that they would come into our own backyard until the pandemic of SARS-CoV-2 or COVID-19. And so even though we expected, this SARS-CoV-19 was really quite unexpected. So the second lesson that I fell back on is a quote from Arthur Ashe, the famous tennis star, where he said, "Start where you are, use what you have, and do what you can." So where did we start? And I think Dr. Barnabas highlighted it really nicely when she sort of drew your focus to that December to March period in 2019, 2020. But really it became real for all of us Saturday, February 29th, when the CDC put out an emergent press release saying that the state of Washington, where Dr. Barnabas was, reported 3 hospitalized patients who tested presumptively positive for the virus that causes COVID-19, including one that died. So until this day in February, we were all watching this unfold on the news thinking, it's not going to come here, and if it does, it's not going to be a problem. So once it hit Washington state, the problem got very real for all of us. And in fact, it got even more real for those of us in Boston when we got a call from the state health department that said Biogen was having an international conference at one of the Boston hotels, and we're now sending 60 patients to Mass General to be evaluated for possible COVID-19. So I sort of fell back on Arthur Ashe, start where you are. And so the first question was, where are we? And so it was March 6th, 2020, and we knew COVID entered Boston. We got notified by the Department of Public Health that 60 people would be coming to MGH for evaluation. We didn't have a test, but we knew the full viral genome was sequenced and it was published. And we also knew that the CDC recently developed a COVID test, but it wasn't yet readily available. It wasn't available at all. And we also learned that we weren't going to get it at MGH anytime soon. So the second part of the quote is use what you have. And so really we had things, we didn't have more things than we had. And so what did we have? The first thing that we didn't have was access to the CDC test. And we knew 60 patients were en route coming to our emergency room to be tested for COVID. We also knew that the CDC test was approved for use on a very specific instrument or testing platform, which wasn't compatible with any of the instruments that we had in our own laboratory. So even though the CDC test was on the horizon, the patients were coming into our emergency room imminently. And so what did we have? Well, for starters, we are a state-of-the-art clinical microbiology lab. So we have incredible instruments like this one, which, by the way, just to put it in perspective, is the size of a Ford F-150 pickup truck. And this platform can do very high throughput molecular diagnostics like a COVID test, but you need to have the test itself, all of the reagents, and all of the supplies. And so you can see on March 6th, we were empty. We had nothing to put in this giant machine. And so even though we had state-of-the-art technology, this was just a giant paperweight because we couldn't use it for COVID testing. So we had the technology, but we didn't have the test. So we really relied on what MGH has in spades. And so what is that? It's really not about the equipment, it's all about the people. So what did we have on March 6th? We had 2 Rhodes Scholars. We had a room full of incredibly smart people who had deep experience developing molecular style tests. And we had a microbiology research laboratory with equipment that could be repurposed to start to develop a COVID test. So we rolled up our sleeves, and on March 6th, we said, help isn't coming our way. So we started to develop our own COVID test. So we made the decision to make our own test on March 6th. By March 12th, really 6 days later, we created and fully validated our own highly sensitive PCR test. And 1 week after we started, we went live with our test. It was on a beat up old research instrument that I had in our laboratory. And we were able to enroll or able to test only 20, 25, 30 patients every 24-hour period. So it was painfully low throughput and painfully slow. But what it gave us was the ability to start testing the sickest among us right away. MGH, because we had this test, enrolled the very first patients in the NIH remdesivir clinical trial. And as you may remember, remdesivir is the most promising intravenous antiviral drug that we use for COVID. And because we had an early test that we brought up quickly, we were one of the leading sites in the entire country to enroll in what I think is one of the more pivotal clinical studies that has been done to date. By April 4th, all of our paperwork was in, and we received FDA emergency use authorization approval for our home developed assay. And we were the 4th center in the United States to receive that kind of approval. So we were really up and running. And we were the first hospital in the state of Massachusetts to be able to test COVID patients. And we worked collaboratively with the Department of Public Health because we had as high a throughput as they did to test COVID patients. So we developed a public health bent, testing people around the state on an as needed basis. And just to put it in perspective, the biggest diagnostic company, Roche, received emergency use authorization approval 2 weeks after we did, developing our own test in-house. So then we come to the third lesson, which is by the great quote by the great basketball coach John Wooden, where flexibility is the key to stability. And so as you all remember, and we're all still suffering from extreme supply shortages that really threatened our ability to do any testing. And what was really interesting was that we were tied to certain commercial vendors who have this lock and key model where you can only use their product on their instrument. So you can only use the Roche test on the Roche instrument. And because of extreme supply shortages, all of these different assays were at times not available for use because of different shortages. So we at the MGH quickly realized that we had to be flexible in order to be stable. And so we quickly pivoted to bringing on multiple test platforms that gave us the flexibility to move back and forth between different testing vendors so that we were never caught without supplies. And at the peak of the COVID pandemic, we had 5 different tests up and running every day. And so when one reagent was in short supply, we would just shift all the testing over to the other 4 platforms. And that really made all the difference in the world. And that got us through the pandemic response essentially unscathed. And the MGH was the only hospital in Boston that never had to divert a test because of supply shortages. So once we were up and running on March 13th, we haven't looked back and we haven't stopped, and we're doing an incredible amount of testing. So just when we thought it was coming to an end, we pull up the quote from the great Yogi Berra. It's like deja vu all over again. And so we dealt with one surge after another. And as you all know, we had the Delta variant, and then we have the Omicron variant. And so it just is deja vu all over again. And really the variant story started in December 2020, when that first alpha emergent, the alpha variant emerged, and that was called the B117. And we used to call it from the countries that they originate, but that doesn't seem fair to any country. So we now call it Alpha, Delta, Omicron, etc., based on the Greek alphabet. So we knew that these variants were potentially very threatening, and they started to emerge every week, every 2 weeks, every 3 weeks. Another one hit the news where we had to start to become prepared. And we also know that in December 2021, the Omicron variant surged, and that was highly infectious, much more so than the other variants. It made our vaccine less effective, albeit still highly effective. And it made certain monoclonal antibodies resistant to, or the virus was resistant to certain monoclonal antibodies. So another surge we learned in December 2020 was imminent. So what did we do? We very quickly had to develop our capability to do whole genome sequencing, where we're basically doing ancestry.com on every viral isolate that comes in the door at MGH. And so now we're one of the leading groups in the world sequencing virus and sharing those sequences with a global database so that people can track and monitor the different viral variants. And so we now do this on every isolate that we have. And again, we're one of the leading contributors. And I just want to highlight one of the really cool things that we're able to do with whole genome sequencing. And that is basically looking at the genetic code of a virus really from A to Z. And so once we developed this capability in-house, we were able to go back to all of the first samples that we had from that Biogen conference. And we sequenced out where those viruses originated from. And furthermore, where did they spread to? And this was a paper that our group led by Jake Lemieux published in Science. And we were able to see that that Biogen event was one of the super spreader events that got the pandemic on fire. And that virus came into Boston from over 120 different introductions. And when we sequenced those out, we found out that they very quickly spread to Florida, North Carolina, Ohio, and Nebraska. And so with that said, we were able to not only track where the virus went, but also where it went to. And we're still tracking it every day. And so that really leads us to the fifth lesson that comes from one of our founding fathers, John Adams, who said, every problem is an opportunity in disguise. And really during the early stages of the pandemic, you couldn't convince me that this problem was any kind of opportunity. But in hindsight, it became really clear that this is our wake-up call, and this is our opportunity to set up a situation where we can be prepared, truly be prepared for any bio threat or any pandemic. So we have a problem. Where's the opportunity? And I think the first opportunity is to be prepared and never let history repeat itself the way that COVID did. We stay flexible. We don't get tied to any one commercial diagnostic platform. And we take advantage of that unique position that the Mass General occupies really at the intersection of clinical medicine, science and academia, public health, and research. And it was that collaboration between all of these groups that are literally right here down the hall that we were able to take advantage of. And so it was that opportunity that helped us solve the biggest challenge that I've ever confronted in my life. And it just really highlights and underscores the need to continue to develop the infrastructure to identify, track, and surveil the full range of infectious diseases, from emerging viral infections like COVID. Today we're dealing with monkeypox, all the way through multi-drug resistant bacteria and other pathogens. So with that, I want to just sincerely thank everybody for all of your help and support, because it was because of you that we were able to do our jobs.
Well, Eric and Ruanne, thanks very much. Not surprisingly, we have a ton of questions that are beginning to come in, and I'm just going to start going through those. And just to remind the audience that as with every 1811 symposium that we have, we also welcome questions about the hospital in general in terms of questions that you might have. So let's just jump to the questions right now. So the first one comes in: how has the collaboration between your departments improved patient safety outcomes for patients and faculty alike at Mass General? So I think both of you should both address that. And Ruanne, why don't we start with you?
Great, thanks so much. This is a really good question, because it shows how important the relationship is between the microbiology lab and clinical practice. The patient case really directs what the lab looks at. And of course, we can't do our work without the laboratory's scientific expertise. And so this very close collaboration means that we can identify for each person who has an infection which antibiotic works. We can scale this up to do research and think about trends, and we can share this information and impact patient care even outside of MGH.
And I completely agree with everything you said, Ruanne. And one of the things that we're really blessed to have at MGH is the infectious disease unit is physically attached right down the hall from the clinical micro lab. So when I'm on the clinical infectious disease service and seeing patients, I have to walk through the microbiology lab in order to get to the patients that I'm going to try to help. And every infectious disease physician stops by the lab, looks at all of the data that we're generating on their patients, is able to tell us, this is what we think this patient might have. Can you help us try to identify that? And vice versa. And so I think the two groups fit together beautifully. And I think the collaboration that we have is allowed us to make, it's really because we have each other that we all rise. And we just really couldn't do it alone.
Eric, we'll stick with you for a minute. You know, in my introductory comments, I used the phrase that pathology really was the invisible engine of the hospital. So I'd be interested, the visibility of the contribution of pathology was just remarkable about how quickly the value of your department became known. And so what was one of the most surprising outcomes that came from your department's collaboration during the pandemic?
I think probably the most surprising outcome is the speed that we were really able to, move the, to get things rolling. And so, yeah, pathology is sort of, one of my colleagues and my department chair, David Lewis, always says, pathologists are like the man behind the curtain or the woman behind the curtain, because everybody has to sort of come to that curtain looking for a diagnosis or an answer, but nobody really sees the person behind the curtain. And that's the role that the pathologist plays. During the COVID pandemic, unlike never before, the pathologist, or in this case, the microbiologist, got completely put under the microscope. Because as you remember, everybody for the first month or two, it was all about testing, testing, testing. And so where's the test? Why can't we get tests? How do we get a test? And so it was really a situation that we've never been in before. And that was surprise number one. But surprise number two was really our ability with our infectious disease colleagues to work together to try to figure out, we have 20 tests today, who gets them, right? And so our ID colleagues had to go around and see all the patients and say, ooh, this person's the sickest person in the intensive care unit. Let's try to enroll them in the remdesivir study. And so we would then prioritize that person's tests so that we can get them enrolled in the study. And so it was so many moving parts, but as two groups handling these moving parts, I think we did the best job that could be expected, or at least I hope we did.
Yeah. Ruanne, you have a really unique perspective that not many of us at MGH have. And what I mean by that is that for over the first half of the pandemic, you were at another outstanding academic medical center. And then in January of this year, with the pandemic still coming on, you arrive at MGH. So two questions I have for you. Any observations on both the similarities and the differences in the way the institutions have responded? And secondly, since arriving at MGH, what are some of the ways that your department plan to continue to partner together to impact patient outcomes?
So thanks so much. it was an unexpected experience to arrive during the Omicron surge, to say the least. And I'll just say that a year, sort of 2 years into the pandemic, when I arrived at MGH, many things were working smoothly. So that was a relief. We had a COVID director, the lab was running very smoothly. We had excellent data. We were able to look at the data and think about how the clinical cases were looking, what the clinical cases were looking like. We understood that hospitalization was much less likely, and then we were able to work on getting treatment to people. And MGH is just fantastic in thinking about what are the gaps and how we can fill them in terms of getting treatment to people. Some of the similarities were really that you need to have people who are experts and who have experience and who can work together to do things quickly. And that was also true of the University of Washington with the Infectious Diseases Division and the Microbiology Lab. And then maybe I'll just round off by saying, arriving during Omicron, something that had my attention and still does, is thinking about those trainees, those fellows who've had 2 years of their training all during the COVID-19 surge, and thinking about what we can do for them, having worked so hard over this time.
Well, thanks very much. So here's another question that I think each of you should have a chance to answer. It seems that for every new variant that comes along now, there are two characteristics. On the one hand, maybe because of the vaccine rate, they seem to be less deadly. In other words, fewer hospitalizations and fewer deaths. But on the other hand, they clearly seem to be more infectious. So where do we go from here with COVID-19? Should we expect to see more variants and waves of infections now and into the future?
So excellent question. The mutations that occur for the variants happen at random. So we don't know for sure with each mutation how it's going to affect transmissibility and the clinical case. Of course, it's to the virus's advantage to evolve to be more transmissible, and each variant does seem to escape some part of the immune. It does seem to escape some part of our immune response, and at the same time, we have immunity both through vaccination and through infections, and both of those things combined mean that there are fewer severe cases and fewer deaths. I expect that we will see new variants emerging, that with each variant we'll need to be on top of the data to understand transmissibility and the clinical impact, and that we'll continue to develop vaccines, and it might be that some vaccines will be quite different from the ones we have today, but we don't know for sure because the mutations are a random event.
And I agree. I think there's just one or two things that I would add. First of all, I do think that between people getting natural infection and so many people getting immunized, that the number of people who have immunity that can protect them from getting seriously ill and dying is far greater than early in the pandemic. The other concept is that, and we learned this through HIV, is that when virus mutates, it always mutates, it mutates randomly, but the ones that take hold have some sort of advantage over the one that it mutated from. And sometimes that advantage may be that you're more infectious, but oftentimes those mutations come at a cost, where the virus may not be as fit or able to cause as serious disease, even though it is more infectious. And so what we're seeing with some COVID variants is that as they mutate, they do become more infectious, but they become less fit. And that's layered on top of a community that has increasing immunity to virus, either through vaccine or through infection. So I think we're doing a whole lot better in terms of serious illness. But I have to say, every day I walk into my lab waiting for that curve to drop in terms of the number of positive tests we have. And it hasn't done it quite yet with Omicron. We're still at around 10% positivity of all the tests that we do.
So, Eric, can you speak to some of the ways in which the use of artificial intelligence in pathology is helping and can help to speed diagnostics and treatments?
Yeah, so I think that's really one of the cutting edge tools that we have in pathology, even though we haven't had a way to do it in COVID diagnostics. But really what artificial intelligence does is it identifies patterns in big data sets. And so the way it's being used in pathology is through pattern recognition, particularly of, say, if somebody had a biopsy and that tissue was looked at under the microscope, AI or artificial intelligence might say, oh, I'm recognizing an abnormal pattern from what should be normal. And so that's really how it's being used and how it will be used in the future.
Dr. Barnabas, I'm going to direct this next one's to you. We certainly heard early on in the COVID pandemic about the perceived lack of funding over the years for our public health infrastructure. From your vantage point, do you think that we're actually beginning to attack that problem? And if so, are we doing it diligently enough?
So thanks so much for that excellent question. For our public health infrastructure, one point to be cognizant of is that we haven't always had our partners at the table as we set shared agendas. So here at MGH, we have an emphasis on building community partnerships of really understanding where the needs are within the community and doing community needs assessments. And so I think that is an area in which we can lead the way when we think about public health. It's a shared agenda with the community. And certainly our surveillance is much better, and there are many more options now for disease surveillance and making treatments and vaccination more widely available. But one area where we have the capacity still to grow and to do better is for shared agendas with communities, and to really rely on those community stakeholders to help us lead the way in terms of things like vaccine uptake and confidence in the treatments that we have, and in accessing testing and what is necessary for health.
Well,
so if I can just add to that, because I've given a lot of thought to our public health infrastructure, and I have to say, I've never been more discouraged than at the start of the pandemic when, everything seemed chaotic, and we went into reaction mode we became reactive rather than being proactive. And so somewhere along the way, we sort of lost our way from being preventative or proactive. And we realized that we couldn't really rely on the typical sources that we had for help, which is why we sort of had to just kind of roll up our sleeves here and make the best of a really bad situation. So one of the things that, I promised myself is that at least my lab would never be caught unprepared this way ever again. And that is because I think when we have that best ability to serve that role in our area and in our region. And so hopefully now things are getting better from a public health perspective and everyone's gotten their wake-up call. But I think we paid the price dearly for sort of that long lack of public health infrastructure for years leading up to the pandemic.
Well, Eric, that's a great lead into this next question. And the person asking the question says, I recognize that hindsight is 20/20, and I don't mean this to be critical or negative, but having said that, nationally, why were we so ill prepared to do the testing that we were, and what have we learned from this experience?
Yeah, I think that question hits the nail on the head. And, you know, it was a very interesting time because scientifically and technologically, we had everything that we needed to be able to act quickly and do a good job. You know, in early January, for the first time ever, we had the entire genetic code of the virus sequenced and published at our disposal. And so, you know, the technology and the tools were all in place, but our response, our ability to really kind of, you know, get off the starting mark and mobilize the industry and government and academia to get the supplies that we need to do the testing that we need, it all just kind of fell apart. And I don't think it was ever together. It was just never exposed in this way. But so the good news is we have the technology. We're in a place scientifically that we've never been before. And now we just need to learn the lessons from COVID and put the resources in place so this never happens again.
Yeah. Ruanne, you know, shifting to a more global, you know, topic, what's your view in terms of either the world's commitment, what I mean by the more developed nations' commitments to reach vaccine equity throughout the world?
So this is an excellent and an important question, because vaccine access in many other parts of the world is lagging behind. And in addition, the production of vaccines is also tapering down. Through COVAX, which is a consortium that includes the World Health Organization, there is an emphasis on supplying vaccines and making them low cost. But what I would ideally love to see in this space is the transfer of technology to low and middle income countries to develop and supply their own vaccines so that they can meet their own demands and really be active in the space of vaccine manufacture, and work closely with their own public health systems. You know, it's not ideal where you are the recipient of a much valued donation. You know, ideally we would transfer the technology, and there would be more capacity for people to, you know, determine the rate at which they make vaccines and match that with their public health rollout.
You know, this is just a personal comment. We certainly do have the perception that the mRNA vaccines are the most effective vaccines out there, and that they also require the greatest care in terms of you know, storage and delivery, meaning that other vaccines that don't require that great care are the ones that are most likely to be distributed. And there is that sense that what we're supplying to underdeveloped nations is a vaccine of inferior quality. It's a, how do you respond to that?
So that's a really excellent point. And, you know, I think that it's not just technology, but it's also the logistics that can be worked out. Actually, I've just returned from South Africa, where the Pfizer mRNA vaccine is widely available, and it's given to everyone over the age of 65, and it's available as a boost now for anyone in the population. And, you know, that has required strengthening of the infrastructure to develop, to be able to deliver this vaccine that has to be stored in particular conditions. But there's lots of gains from strengthening that infrastructure, because it means that probably other health technologies can utilize them as well. So I would like to see not only the transfer of technology, but also the sharing of infrastructure and logistics for delivery.
So here's a question that's come in, and actually I'm going to join you as a panelist here in terms of answering it, and I'd like you to answer this question not so much as a scientist or not so much from your scientific expertise, but as a manager or a leader of your department. And so the question is, how is Mass General and its employees doing today? Is the hospital well positioned for future pandemics and outbreaks similar to COVID-19? So let me answer this sort of globally from the hospital's point of view. COVID itself as a disease and treating it disease was our main focus, but there's been collateral damage as a result of that. It's really hard to imagine the stress of really working, continuously for 2+ years, in large part because at any one time there were significant numbers of our workforce that were out as a result of COVID illness, or they were just out because of a need to quarantine for 10 days. And so normal vacations, couldn't be kept. And the healthcare industry was not immune to the, quote, great resignation when people were stepping back. And so I think maintaining morale at this institution and being able to continue to deliver care at the highest level is something that is challenging us. We're certainly investing in programs in terms of well-being, but exactly how we're going to get through this when even today we have as many as 40 beds closed at the MGH because of staffing shortages of individuals who are out with COVID. So I'd say we're doing okay, but we've got a long road ahead of us. So Eric, why don't you talk from your position in pathology, and then Ruanne from your position in infectious disease?
Sure. Just to add what you said, I think the laboratories have been also very hard hit with people getting COVID and having to quarantine. And so when you have a limited number of people and 3 or 4 are out at any given time, everybody else has to do double duty. And I think to be able to do that over a day or two, nobody would blink at that. But it's been 2 years now. And, every day I walk through the lab and you just see everybody's just exhausted. Nobody's complaining, but everybody's just flat out exhausted and just desperately wants this to go away. And then it becomes really frustrating for people to leave the hospital and go out, and it seems like nobody outside really remembers what COVID is. And so it's kind of this paradox of, coming in every day and sort of doing battle, and then going out into a world where, the world has seemingly moved on. And so it's been tough.
Certainly in infectious diseases, we are going through a process now of trying to recover. I'm talking about we are actively entering a recovery phase from the exhaustion that was COVID, and thinking about how we can, even though the future is uncertain, find joy in the future. Remember that we love the dynamics of infectious diseases. We love the detective work and solving those complicated problems. But it is a time of transition for us as we are recovering from really just over 2 years of hard work and facing our 3rd year of the pandemic, and thinking about how we can focus on the wellness of individuals who work with us and continue our science and patient care and training.
Well, I think we've reached the end of the hour, and unfortunately, we have a number of questions that are still out there that we haven't been able to get to. On behalf of the entire 1811 Society, Ruanne and Eric, I'd like to thank you for what was just a terrific program. And more importantly, I'd just like to thank you for what you and your staff are doing each and every day for that next patient who crosses the threshold. And for our donors and participants today, thank you for joining us. And we can't wait until the time that we can get back together in person. So please be safe. Be careful out there, and thanks for joining us. Bye-bye.
Bye.